Good morning, my name is Michael Gillings. I'm an evolutionary biologist from Macquarie University in Australia, and the first thing I'd like to do is to thank Mary for inviting me to make this presentation to you today. What I want to talk to you about is called microbial dark matter by many people. One of my interests is how the hidden world of microorganisms moves around the planet, and what that might mean for the planet and for humans and for the plants and animals that we depend on. So it should come as no surprise to any of you that humans are now the most important and powerful evolutionary force on the planet. We have significant effects on every part of the planet, the atmosphere, the oceans, the hydrosphere, and also on all of the other organisms on the planet. There is no place on the planet that is not affected by human activity. So we have to start thinking about what kind of world do we want to live on. We have an opportunity over the next decade or so to make decisions about the kind of planet that we want to leave to our children and grandchildren, and to make that happen. So we affect all aspects of what's called Earth's critical zone. Now this zone starts basically at the top of the trees or the atmosphere, encompasses the soil and the plants and animals that are living on the surface of the planet, the soil profile, and down through into the lithosphere, the rocks underneath underneath all of the soil. So that's called the critical zone, and it links atmosphere, biosphere, hydrosphere and lithosphere, and this is where soil, water, air, organisms, climate all interact to generate the conditions that allow us to live on the planet. These conditions are often called ecosystem services, and these are things that the planet and the organisms that live on the planet supply us for free. We need to understand these interactions, and that in turn is going to require interactions between all sorts of different areas of human investigation. The critical zone itself is a concept that arises in the geological sciences, and it's time for biological sciences to have input into that area of investigation. So many of these critical zone activities are actually driven by microorganisms. In 1997, Costanza first valued ecosystem services in terms of how much money would it cost us to replace the services that organisms provide on the planet. And the answer was, it's a lot. It's actually $33 trillion, which at the time vastly outstripped gross domestic product of every country in the world. The kinds of services we're talking about here are things like atmospheric regulation, nitrogen fixation, cycling of oxygen, carbon, sulfur, phosphorus, waste control, biological control of diseases, pollination, plant growth, soil formation. All of these are critical ecosystem services that organisms interacting with the critical zone infrastructure, the soil and air and water, they produce this for us automatically. Microorganisms have a central role in most of those things, and yet we all go through our daily lives not even thinking about microorganisms and what they're doing. So my question is this: how does human activity on the planet influence the distribution of microorganisms, their abundance, and their activities? So how do humans affect microbial activities, distribution, and abundance? I'm going to give you some examples. We don't have time to go through a lot of different examples. The first one I'm going to talk to you about is how microorganisms get spread around the world. So prior to humans dominating the globe, microorganisms only moved passively. They moved via water flow, via wind, and other physical movements. That has all changed with humans. For instance, here's one example, ballast water in ships. On the right-hand side here we've got shipping lanes, and you can see the entire world is networked with literally thousands of shipping lanes. All of these shipping lanes have large cargo vessels travelling on them that pick up ballast water. Those vessels pick up water from one place on the planet, steam to another place on the planet, and release the water. It's difficult to tell exactly how much of this happens, but we do know that 100 million tonnes of ballast water is released into US ports every year. Now, people have thought about transport of mussels and invasive starfish and seaweed and things like that, but virtually no one has ever really thought about the huge numbers of microorganisms that must be transported around the planet that way. We do have specific examples where cholera has been transported, or toxic dinoflagellates. We know about those because they've caused an immediate human health problem. But microorganisms are being transported to places they've never been before, and all of those microorganisms have ecosystem services and things that they do. So are we actually disturbing or perturbing those activities? The other thing that you might not realise is that humans now move more soil and rock than natural processes combined. So all of the erosion by rivers and glaciers, humans move more soil and rock than all of that. And of course soil is full of microorganisms. So is rock. Not as many microorganisms. Soil has about 10 to the 9, about 1 billion per gram. Rock has far fewer, but that moves organisms around the planet to places where they ordinarily would not be. The next thing we have to think about is the kinds of microorganisms that are associated with animals. So every single animal is host to large numbers of microorganisms that are essential for those animals' health and well-being. And this, to me, as it says on the title of the slide, is the world's scariest graph. So let me just explain what this means. 10,000 years before the present, this is the number of terrestrial vertebrates, so things with vertebra on land. This is the millions of tonnes, so about 200 million tonnes. The graphs on the right-hand side show, as we go from 1900 to 1950 to 2000, what happens to those wild animals. And you can see the green slice here comes down to a tiny little sliver, and by 2050 it's hardly visible on this graph at all. On the other hand, the blue part of the histogram, that's the biomass of animals that humans eat. And the red part of the histogram, that's the biomass of humans. So in other words, by 2050, we're looking at 98% of terrestrial biomass of vertebrates being humans, pigs, cows, chickens, and sheep, essentially. Now, that's terrifying, but it also has microbial consequences, because each one of those host organisms has its own specific microbial flora. So in other words, what we're doing is we've completely altered the microbes that are being deposited in feces around the world to being dominated by the microbiota of just those essentially 5 species. The consequences of that, we don't really know what they are. All of these organisms are released sometimes into wastewater that is treated, but the vast majority of wastewater is actually not treated at all. So a lot of places around the world rely on wastewater for agriculture as irrigation. 80% of that irrigation is not actually treated at all, and so we end up with all of these microorganisms being spread onto agricultural fields, where they then cross-contaminate food and animals grazing on those fields, and those animals and food are then distributed globally. So we've got this situation where we've vastly changed the dynamics of organisms that are being released into the environment from animals, and we've set up a situation where there's a cycling of these organisms back into human and animal ecosystems because we distribute food globally and consume it globally. The other thing is, where we have wastewater treatment plants, where wastewater is treated, we put all of the organisms into one big vat, essentially, that has enormous microbial abundance and is also full of selective agents. Agents that ordinarily should not be in water. Pharmaceuticals, antibiotics, disinfectants, heavy metals. All of those things are in that water. That sets up a hotspot for bacterial evolution, for organisms to acquire resistance genes and swap resistance genes, and for new combinations of genes to appear. So, how can we monitor these processes? One of the ways of monitoring this, it's kind of difficult to track individual bacterial species, but there is a way of thinking about connections between locations and thinking about how large this problem might be. One of those is to use antibiotic resistance genes themselves as proxies for the movement of species around the planet. Genes are easy to monitor, and there's a great deal of interest around the world in monitoring these genes because of their human health impacts. Where did these genes come from in the first place? Well, to start with, these genes are present in the environment, and as we have started to use compounds to control bacterial growth, bacteria in our gut have picked up resistance genes for these compounds, and as a result, many organisms that live inside humans and inside our domestic animals now carry antibiotic resistance genes. The original source of those was the general environment, but with the widespread use of antibiotics, exposure to disinfectants and heavy metals, basically all humans now carry resistance genes in their gut bacteria, and the numbers of these and diversity increases as we age. So children to school children to high school students to adults, the number of different resistance genes that we carry increases. What that means is that humans and their agricultural animals that are also treated with antibiotics are now a major source of resistance genes. So those resistance genes come out in feces and waste and go into the environment. How bad is this? Well, it's pretty scary. This is a study that we did along the coastline of China, looking at estuaries along 4,000 kilometres of Chinese coastline. And bear in mind, these estuarine sediments should not have any antibiotic resistance genes in them. They probably did not have any antibiotic resistance genes in them 200 years ago. Now we find that every single sediment is polluted with resistance genes from probably agriculture and human antibiotic use, and those numbers range from 1 million to 100 million copies per gram of sediment. So bear in mind that lots of people around the world depend on foodstuffs that utilise estuarine sediment. Shellfish, crustaceans, fish, all of the things that use near-shore marine habitats feed in or around sediment, and that creates, again, a circle coming back into humans. What this does is it makes resistance genes spectacularly successful and makes them able to cycle between environmental compartments with ease. So humans, our domestic animals, our farmlands, all cycle resistance genes round and round. Probably the best example and the most spectacularly successful example of this is not actually a resistance gene, but it's a gene that vectors antibiotic resistance. It's called the class 1 integron. It's something that I'm interested in. And I decided to try and look at class 1 integrons, which have only really appeared since the early 1900s as a common gene, and they've done so because they're given this huge advantage because they're able to carry antibiotic resistance genes along with them. So we can do some quick calculations, back of the envelope calculations. You look up people who've investigated the abundance of these, and we know the abundance of these class 1 integrons per gram of feces. We can calculate the numbers of grams of feces that each of our crop animals and humans produce every day. We know the size of the human population, and the end result is that somewhere around 10 to the 23 copies of these genes are released every day. So that's a really big number. And that results in significant environmental contamination. We can actually track this through the environment using a whole series of studies that have asked the question, how abundant is this in environmental samples? And so this plot here just summarizes 45 different studies on various environments, and the main point here is that this red line across the top represents one copy of this gene per bacterial cell in these environments. These are just the average stats, but if you look at the individual studies themselves, it tells you something. The place where there's the least of these genes is an Antarctic soil sample, and this may represent the original and, if you like, natural abundance of these genes around the world. Soil samples are elevated in the numbers, but then as soon as we get to farm manure, these numbers go through the roof, and the highest levels are actually found in chicken manure. But as we take this manure and put it into wastewater, you don't see any decline. As wastewater is treated into sludge or into effluent, which is now treated wastewater, being released from wastewater treatment plants, you actually don't see any decline in the abundance of these genes. That's because wastewater is designed not to remove genes or pollutants, but to remove organisms. So the genes actually make it through the treatment. And then, because this effluent is going into rivers and lakes, the number of this gene in rivers and lakes is also elevated, hugely elevated from pre-1900, let's say, levels. Once it's in rivers and lakes, these genes bind onto freshwater sediment and appear as sediment, and the worst places in terms of numbers are actually downstream from pharmaceutical plants, because pharmaceutical plants release waste antibiotics into the environment. So, these genes even make it into our own homes. This is some work I've been doing with vacuum cleaner dust. So you test vacuum cleaner dust, and this is actually from my house. So this gene being detected in my own home. So it's everywhere. These genes are obviously significant pollutants, and we're now starting to think about genes and microorganisms as being pollutants. So they're present in diverse species, but they're also maintained because, don't forget, these genes are all attached to each other, and they supply resistance to metals and disinfectants and antibiotics. Unlike other pollutants, they can replicate. So if you think about pollutants, there's conventional pollutants, carbon dioxide, heavy metals. There's novel pollutants that humans make, radiation, xenobiotic compounds. And then there are these pollutants that replicate, like the genes I've been talking about and invasive species. But these interact. They interact because conventional, xenobiotic and invasive species all increase the numbers of xenogenetic pollutants, these genes that are of interest here. That resistance spreads into wild animals and into environments, and also spreads even into conservation. So that where we try to have captive breeding systems, we still find that these genes are making their way into these organisms. So, consequences for humans. Obviously, there are health consequences in the fact that resistance genes lead to organisms that can't be controlled in medical contexts. So, each one of us has about the same number of bacterial cells living in us as we have human cells. But during modern civilization, the diversity of those genes has declined. We know that because hunter-gatherers have many more bacteria in their gut and more diverse bacteria than we do. So that some bacteria are completely missing from humans in developed society. We don't know the full extent of consequences that might arise from that, but we do know that there are lots of things that we do that affect our microbiota: caesarean birth, bottle feeding, antibiotic use, our diet, our own genetics. And we need to care about this because a healthy microbiota is essential for our immune function and for our general health, but also for our mental health as well. So, in the Anthropocene, the age of humans, microorganisms have been significantly affected by human activities. Adverse changes to the human microbiota, significant changes to the distribution of microorganisms and their abundance, and the abundance of the genes that they contain. We have changed the relative roles of bacteria in cycling nutrients around the planet, and we've driven rapid selection for antimicrobial, metal and disinfectant resistance. Thank you.